Now we get to the issue of bioavailability, and biochar is one of these things I've just trotted out as part of that discussion. But bioavailability is a big issue with regard to organic contaminants and to bioaugmentation, this business of adding organisms to enhance the decomposition of materials. None of those magic organisms will decompose anything if it isn't accessible to them. Metal contaminants, another area where the availability of that contaminant to plants, to humans, is a big issue, and it is controlled by the soil matrix. Carbon sequestration, and biochar, of course, is important here, is a big one. And we'll just stop on that for a brief moment, because one of the goals that people talk about is let's make biochar, let's add it to soil, it'll stay forever, and we will have sequestered carbon, and that will then allow us to remove some of the carbon from the atmosphere. If that works, that's good. And the story isn't yet fully told, but it's something that does deserve taking a peek at. But it all comes down to, one, availability, and two, of course, decomposability as well. Trace elements, biofortification. But biofortification has started to poke its head above the water, and is an intriguing idea. Now, it has a couple of manifestations. One is through the fortification of foods and things. The other is through the growing of crops that are enriched in certain, either certain elements, zinc, copper, iron, for example, or in certain vitamins through plant breeding, developing more vitamin-rich plant varieties. And I read on the plane, actually, yesterday, a very interesting article I think it was published in Science not too long ago by Dr. Ramaswamy, who had conducted or led a group dealing with this. And his argument is that there's about 870 million hungry people in the world, that there's hidden hunger, which is beyond simply not getting enough energy and not enough protein in your diet. It's also a diet that lacks vitamins and various essential other trace minerals. And he was arguing that it is indeed possible to develop plant varieties that will have increased concentrations of trace elements in them, that gets into the seeds or the part of the plant that is eaten, and of course gets eaten and subsequently absorbed. There's a whole bunch of steps along that line that are needed to make that thing work. But if it does work, and if those plant varieties are in fact accessible to people who are existing in a subsistence agricultural mode, and many, many millions of people are, then it's a big boost to the health of the world community. But it's also an important message respecting soils, because soils are the source of those trace elements. And whether that will modify what we do or not, I don't know, but it's certainly something to keep our eyes on. And Ross, of course, doesn't seem too keen to start adding trace element fertilizers. So I'm not selling them, Ross, it's just keep at it and keep your eyes open for these things coming along the line. The other issue is questions about shifting climates and shifting biogeoclimatic zones and shifting soil microbial communities. Quite an interest there, partly through the business about mycorrhizal associations of plant roots. And these are our maps. The top left map is simply the currently mapped biogeoclimatic zones. The next one is a prediction of them to see whether the model worked. And then the next is 2001 to 2009. They're all pretty much the same. You see a big blue wad in the middle, subboreal spruce. And then if you move to the bottom line, you have 2011 to 2040, 2041 to 70, and 71 to 10. And you'll notice a gradual loss of some of those zones, moving north of others. And of course, in the northern part of the province, things are starting to appear that didn't appear before. So the question then arises whether or not the soil microbial communities that may be important for the survival of various plant species will move with these moving geoclimatic zones. And I've just used BC as one example. There are others. It's interesting because of the variety that exists in BC, both with latitude and with altitude. And so, of course, the mycorrhizal community thing, some of the questions being asked there. Simulation models of microbial physiology to understand nutrient cycling, part of this other microbial business. And this particular paper had to do with modeling the change in microbial carbon utilization efficiency as a way of dealing with changes in temperature optima of microorganisms. And then detailed microsite habitat information that you can get through nanomicroscopy and such techniques, which will give you very good information about the chemistry of microbial habitats. Moving into, you'll notice we're getting farther out into that sort of molecular biology area. Novel genes, enzymes, organisms, and consortia. The rhizosphere microbiome and its manipulation is thought to be a mechanism to help suppress plant disease. We've thought that for a long time. We've got better tools to deal with it now, and there's a revival of interest in that subject. Antibiotic-producing microbiomes, of course. Who knows about Selman-Waxman? Aha. What do you know about Selman-Waxman?
He discovered antibiotics in Sanborn Field,
I didn't know it was that field, but I will not argue with you. Whereabouts in the field? In the crops?
In the soil.
In the soil, right. Got a Nobel Prize for it as well. And that was the first record of antibiotic production and the organisms producing it. And of course now the whole world's full of antibiotics, maybe to our detriment. But there is still this search for antibiotic-producing microbiomes or consortia of microorganisms. And where do people go looking for it? They go looking for it in soils. Novel cellulitic and hemicellulitic genes and enzymes. These are really important to the whole bioenergy industry. One of the big problems that faces the commercial production of ethanol from lignocellulosic wastes is the difficulty in fermenting those materials. Starch is easy, but cellulose is difficult, and it's even worse if it's attached to lignin. And so that enterprise is also digging about in soil to seek these novel enzyme systems that may in fact help open some doors to a more economically viable way of doing that. Whether you or I think that we should be producing ethanol from lignocellulosic wastes is a totally different story. It is happening, and more of it is going to happen while there's a need for a portable liquid fuel. Just a couple of quotes about this subject. Soils are considered to be the most diverse microbial habitat on Earth. So it's not just the soil brown zone or the brown soil zone, it's any soil. Once you get inside it, it's full of species, a vast array of them, more diversity than anywhere else on Earth. And I torment my biologist friends by telling them, don't worry about some of these ecosystems. We've got soil microorganisms and we've got the soil habitats, and they'll look after us all. Of course, if you think back to what they have done to the world, the globe over a few billion years is pretty astounding. The Earth used to be a totally anaerobic, reducing environment. Along came oxygenic photosynthesizers and now killed off all those others, and here we are. So they have some power over the world, and they will survive. So I'm not biased. Soil metagenomes, the sort of large genome pool within soil, represent an unlimited source for the discovery of novel biocatalysts from soil microorganisms. And here I want you to start to ask yourself, my 45 minutes aren't up yet, ask yourself, is this an example of people using knowledge of soils? Or is this just an example of people going out and getting a bucket of dirt? And the more I think about it, the more uncomfortable I get. So I'd like you to get uncomfortable too and think about it. And here we get this integration, the next question that I think is important, integration over orders of magnitude in size. And Dan, I actually went and reread your paper from 2003, I believe. That's a really good paper. Why isn't Dan giving this talk? So I hope I am not taking anything in vain here, but I've long been fascinated with this problem of scale, because we've got, in going from our size scale, which is about the meter size scale, and some of us are closer to that than others, to the microbial size scale, 6 orders of magnitude. Which, if you think about it the other way, if we go from the meter size scale up 6 orders of magnitude, you're talking about 1,000,000 meters. And if you think about going from the microorganism down to submicron size particles, you can account for another 2 or 3 or 4 orders of magnitude. And if you're thinking about going up another 2 or 3 or 4 orders of magnitude beyond that, you're out of the solar system. So it's a very big scale range that we need to deal with, and it's fun to deal with it, but we don't have very good tools yet to do that. And we have processes that are important and manifest themselves out in your fields. Denitrification, for example. It occurs at a site that is about 1 or 2 square microns in size. Many of those sites. But you see it out in your field, which is meters or hectares in size. How does that happen? And how do we understand what is controlling that microsite process and its manifestation at the macrosite scale? And do you use ecosystem properties? Do you build it up, from little to bigger, big, like onion skins and so forth? I thought a lot about it. Many people have thought a lot about it. I'm not sure we've got the answers all worked out just yet. One thing I'm pretty sure about, though, is that the minute we start to play with spatial scales, we're also playing with temporal scales. You can't really separate them. Connections between soil biology and soil physics. They don't seem connected, but they're fundamentally connected. And this is just a little diagram of diameters of pores going from 0.1 micrometer to 1,000 micrometer, and the distribution of those pores in cubic centimeters per cubic centimeter in a sand or a clay soil. And you'll notice that, of course, there's more of them in clay than in sand, as we all know. But the interesting thing here is the 2 vertical bars. Those 2 vertical bars represent the window within which soil microorganisms can likely inhabit soil. The space above those bars is too dry for them to be in. The space below those bars is too small for them to fit into. So it's only the goings-on between those 2 bars that you can actually envisage soil biological activity and biochemical activity taking place. Earthworms throw this all out the window because they come along and ingest the whole mess and mix it all up. So that's a different story, so don't confuse what's happening with large organisms like earthworms that move the soil about and what happens with bacteria and fungi that don't move the soil about. Carbon storage, big issue. And you'll notice in this thin section, nice black Chernozemic soil, black carbon around the particles, spaces in between, carbon is stored, and so we have a pretty good mental image about that. And that's important to bioavailability, to the physics biology slide and connection. It's an important attribute with regard to stable structure. Reducing toxicity of xenobiotics. If they're stuck away where nothing can get them, they can't do any harm. Reduce migration of xenobiotics and viruses. Again, if they're stuck away in nooks and crannies and they can't get out, they can't move. And if they don't move, they don't usually do any harm. And carbon sequestration, where we want to store carbon away from keeping it out of the atmosphere. However, it may not be desired if in fact you're wanting to remove contaminants. And so the soil reclamation and remediation fraternity has this really fascinating problem on its hands. On one hand, if you can get these materials stored away in soil in a really protected fashion, they're no longer a problem. But if they're stored away in this protected fashion and you have to get them out, you have a problem. So it can either be a good thing or a bad thing depending on how we approach that, and the role that bioavailability and no bioavailability in the toxicology literature is rife with arguments. So we're not going to go there today, but it's a very interesting conundrum. Is this desirable? Is it not desirable? And it's fundamentally a size, location, reactivity issue. Hopefully we'll resolve that in the future. The users are agronomists. Sorry, Ross, I too have the Breton plot slide. Thank you, Jim, and I think Cesar Izzarelli for making this slide possible. And a picture of the Luvasolic profile beside it. This is fundamental to some of the biggest single issues facing soil science and the world into the future, period. All that talk about food, fuel, fiber is talk about agronomy. And we'd better not forget it. Biologists and ecologists love it. Where would we be without our mushrooms? And here, of course, is a group from Switzerland, actually, in a forest in BC, south of Prince George, studying ecology and so forth. Epigenetics, the concern about possibilities of methylation, for example, of DNA that can alter the activity of the DNA on into the future, and the growing observation that there may in fact be ways of monitoring that using earthworms in soils to monitor their DNA methylation. Still in its infancy. Where it's going to go, I'm not totally sure, but epigenetics is up and running on a tear right now. Metagenomics is also up and running on a tear, but it really addresses the whole muddy mess that's there and doesn't try to separate things out to individual species. It just says, let's look at what's there and get after the genes that we need. Civil engineers, who's here from Edmonton? Good. This is the Edmonton-Bedminster reactor, which I hope is still working. And way back in my other life, I was involved with Jeremy Leonard and a few others doing some composting work prior to the implementation of this process. So I was a soils person, so I claim that soils has a connection to the Bedminster reactor through that connection. And of course, it produces compost, and we all know where compost goes, and I believe in this case it's being used for mineland reclamation. Whether that's still the case, I'm not sure. And ecotoxicology, of course, a big problem, and earthworms, of course, as you see in the slide, part of that discussion. Other points, but are they really using soils? Exo-biology is fascinating. You know, this whole business about searching for life on Mars and all the other things. Really, they're searching for evidence of soil formation. You can't form soils without life. They're looking for evidence of soil organisms, bacteria specifically. And this is a photo of Mars, taken from the Mars Pathfinder some years ago. I think it's a 2006 photo, I'm not sure the exact date. But you can go to the source and get more of them if you like. It's quite fascinating, rocky. It's like southern Alberta after the Dust Bowl days. You can use this one too if you like, Ross. show what it could be like if you don't mind your ways, folks. So we get to this Quo Vadis business and the unified soils knowledge, and anybody who knows me knows I cannot resist this slide, no matter what I'm talking about. So here we go with the lithosphere and the hydrosphere and the atmosphere and the biosphere and soils. And that to me says volumes. That's what it's all about, folks. It's that integration of stuff. And soils are there, and they unify all of those pieces. And they inform all of those things. They alter the atmosphere, they alter the hydrosphere, they support the biosphere, and they alter the lithosphere. So, are we going to have our nice rippling system? Or is it going to fade away to be replaced by something not so well defined, not so substantial, and fragments all over the place? And I would ask you to think about that, because I see that happening. And that's not a happy thing to see happening to a person who really believes that that unified understanding of this unifying material is critical for humanity. But it doesn't have to happen. And if we develop an appropriate communication and dialogue between us in the middle, if you will, and all other users, such that what they gain comes back to us, what we know goes to them, it seems to me everybody's going to win, and we're going to have a future that is going to be really useful. So those aren't bad things, those are good things. Those are the bits that are going to make us vital and alive and have a future, but we have to have a good conversation with those other folks to make that happen. The choice is ours, the future is what we invent. And I think our future that we should invent is here. I think we got to get back to thinking about always having in our heads this image of a pedon and the landscape it lives in, if we're going to keep things moving forward. And that is it. We can't invent those things, but we can certainly organize our thinking to respect them and to value what they do and to value the understanding of what they do for humanity. Thank you very much.